Okay, so I'm going to start the timer because I want to make sure I don't go over time. So thank you so much for the invitation. I also apologize for not being there in person, but I really appreciate the possibility to join remotely, which is really fantastic. So the title of my talk is Molecular Mimicry, Ecology, Evolution, and Applications of Doppelganger Peptides from Cone snail venoms. And I first want to say a little bit about molecular mimicry and what we refer to as doppelganger toxins these days. So molecular mimicry, as opposed to morphological mimicry, where one organism mimics morphological traits of another, refers to the mimicry of molecules or of at the molecular level. And in the case of doppelganger peptides, let me see if I can turn my pointer on. Okay, I hope this works. So in the case of doppelganger peptides, we're interested in toxins. So if you imagine all the toxins that are out there in the venomous world, we're interested in these toxins that mimic peptide hormones or neuropeptides. So it's a subset of toxins that we're interested in that we have started to call doppelganger peptides to give it kind of a unified framework. And the reason I'm interested in doppelganger peptides in our lab is that it's basically based on the discovery of insulins, first in Conus geographus and since then in many other cone snail species. And these are doppelganger peptides of the endogenous hormone insulin that is made in fish and human and almost all other organisms. But arguably the most famous doppelganger peptide now is xanthine-4 from the venom of the Gila monster that mimics the human hormone GLP-1, glucagon-like peptide 1. And it was the first GLP-1 analog to be approved for the treatment of diabetes back then and now obesity before other GLP-1 analogs entered the market. Doppelganger peptides are not limited to venomous or poisonous animals. They're actually found broadly in other conflicting interactions, such as in parasites or pathogens. So pathogens that infect rice, for example, make doppelganger peptides of plant hormones. And they have also been reported in sexual interactions, which is something I won't cover today. So for today's talk, I mostly want to focus on venoms and on the venoms of cone snails. But recently, Thomas Koch in my lab and I looked at, wanted to survey what's the diversity of doppelganger peptides in venomous animals. And this is the rather complex figure shown here. So I want to make sure that I take you through it because I'll come back to this figure throughout the presentation. So I first want you to focus on this yellow triangle, and the yellow triangle depicts where we find specific neuropeptides and peptide hormones. So for example, in metazoan, which are all multicellular organisms or animals, we see insulins, for example, RF-amides and EGF hormones widely distributed. And then in bilateria, which is basically almost the same animals with the exception of non-bilaterian organisms such as cnidaria, we see other additional peptide hormones such as tachykinin and vasotocin and so on. And then in deuterostomes, which is chordates and echinoderms, so it includes us, we have families of peptides and hormones, neuropeptides and hormones that are only found in deuterostomes, such as bradykinin or GLP-1. And then finally, in protostomes, which is this group shown here, we see additional peptides such as myosuppressin and proctolin and so on that are found there. And then in cnidaria, we have a small subset of neuropeptides that are specific to cnidaria. So now when we look at the doppelganger peptides, so these are basically toxins that mimic endogenous peptides that have been reported across the venomous kingdom, we see that they're actually broadly found. And I know there are a lot of snake people in the audience. So for example, in snakes, it's been long known that various species of snakes make natriuretic peptides, and some snake species also make endothelins. We have some people that have worked on fang blenny venom, which is really beautiful studies that have shown that fang blennies or venomous fang blennies also use doppelganger peptides of neuropeptide Y and also endorphins, which are opioid-like peptides. But for the purpose of today, and actually most of the work from our lab has focused on these peptides from cone snails, which are marine snails, as we just heard. And as you can see here, wherever you see, again, a blue triangle, we see the presence of doppelganger peptides in these venoms. And as you can see, cone snails have a lot of these peptides, and we're continuing to discover more. And it could partially be due to their biology, but it could also be because we're in the cone snail field have looked for these peptides more systematically. Okay, so when I submitted the abstract for this meeting, I basically said I would talk about the ecology, evolution, and application of doppelganger peptides. As I was putting this 15-minute presentation together, I realized that that was a little ambitious. So I'm just only going to give you kind of a high-level overview of what we're doing in terms of ecology, evolution, and application, and I want to start with application. So it turns out that these doppelganger peptides are really useful drug leads. So they're very useful for biomedical application. And this is due to their unique evolutionary history. So if you imagine neuropeptide and peptide hormones, many of them are implicated in various diseases, but they're actually very poor drugs. And this is because they evolved to function in endocrine and paracrine signaling, which means that they don't have to be very stable. They actually shouldn't be very stable. They often don't have to be very specific for subtypes of the same target because they're released close to the site of action where the target is expressed. And they're also not often very bioavailable because they're released endogenously and they don't have to cross various tissue layers or the extracellular or survive in the extracellular environment. However, and this is early on, we saw that with the insulins and many other peptides, doppelganger peptides since, these doppelganger peptides have a lot of these drug-like properties that we're interested in drug development. So as all of you know, toxins are very stable typically. They evolve to survive in the extracellular environment. They're bioavailable. They can cross multiple barriers within the body. And they're often also subtype selective because they're administered often systemically and need to still find the right target of action. And so, for example, in the case of insulins, and I won't go into details for the insulins here, but we reviewed the work we've done on the insulins about 3 years ago. We found that they are very bioavailable. They're fast acting because they're monomeric molecules, unlike human insulins that are dimeric and then also hexameric in order to be stored in the pancreas. And coming back to the Gila monster, exendin, the reason why exendin 4 became the first GLP-1 analog for the treatment of diabetes is because it's actually very stable, and that's due to these 3 amino acids at the N-terminus that make it resistant to proteolytic cleavage by the enzyme DPP4, which human GLP-1 is very rapidly cleaved. So I would encourage people, if you do find a doppelganger peptide that mimics a human hormone, to look for unique properties that doppelganger peptide might have when compared to the human hormone itself. But what is really fascinating, and that has kept me interested in this field of research, is that while we explore the application, biomedical applications, we also learn a lot about the ecology. So doppelganger toxins or doppelganger peptides harbor a lot of ecological information. So they tell us about biotic interactions between the animal and the prey and predators. So for example, the insulin, we showed that it's used to induce hypoglycemic shock, so low blood glucose levels in the prey. And that's a really kind of neat mechanism for prey capture. And then more recently, in my postdoc, Ho-Yan Jung, last year published a paper where she showed that insulin is not the only doppelganger toxin in cone snail venom or in Conus georgaphus venom that targets glucose metabolism or glucose homeostasis. There's actually another doppelganger that is involved. So I quickly want to show you how that works. So insulin from Conus georgaphus venom, or insulin actually in our own pancreases or in fish pancreas, induces lowering of blood glucose. So this is blood glucose level shown here. And if insulin is released or injected, or it lowers blood glucose levels. And if blood glucose gets too low, and some of you may have experienced this, you can experience something that is called hypoglycemic shock, which is defined by being really dizzy, and in extreme cases can put you in a coma and you could die. And this is not a good place to be. But the pancreas actually has a counteracting mechanism, which is it releases glucagon. And so that mechanism of insulin-glucagon interaction is very well conserved in vertebrates. And when glucagon is released, glucose levels go up. So for some time, we thought that the cone snail, Conus georgaphus, probably had a mechanism of inhibiting glucagon secretion to really sustain hypoglycemic shock. And Ho-Yan Jung identified this peptide as actually a somatostatin doppelganger. So it's another doppelganger peptide of a very important human hormone that is released by the pancreas and can inhibit glucagon secretion. So the combinatorial use of these two peptides induces this sustained hypoglycemic shock. And combining ecology with application again, of course, the somatostatin doppelganger also has some really neat properties. So this is human somatostatin. It actually looks the same in fish. This is the consomatin PG1 from Conus geographus. And this is a man-made drug approved for the treatment of various somatostatin-related disorders. And you may appreciate, just by looking at these two, how similar the cone snail venom peptide is to a man-made drug. And the similarity spans this minimized disulfide scaffold and also a D-tryptophan within this loop that is very important for stability and also activity. So millions of years ago, these cone snails already came up with a solution for how to make somatostatin-based drug analogs. And we're interested in this particular somatostatin doppelganger for therapeutic application. So we're doing structure function studies in the lab right now that is not published, but here you see the cryo-EM structure of this peptide bound to the somatostatin 2 receptor, which is its biological target. And then, interestingly, we find that cone snails, many other cone snail species make consomatin-like or somatostatin-like toxins. And for example, this one here does not target metabolism. So coming back to ecology, it actually targets pain circuits through activating a different somatostatin receptor. So it's very specific for a different somatostatin receptor subtype implicated in pain. And we're developing this peptide as the potential drug lead for pain, and founded a startup company last year to pursue these efforts. So ecology and application is kind of nicely combined in these doppelganger peptides. Okay, so insulins up here and somatostatins here are not the only doppelganger peptides that have been investigated for therapeutic application. I already mentioned XNN4, the GLP-1 peptide from the Gila monster. And then there are also some snake natriuretic peptides that have been really interesting drug leads and have served as scaffolds for making analogs between the human and the venom peptide. And then there's another cone snail peptide, which is a neurotensin-like peptide that has been investigated for pain. So I would encourage you, once again, if you find a peptide that mimics a deuterostome or a human hormone, I'd encourage you to investigate whether it has some unusual, interesting properties for drug development. Okay, very quickly, in the last couple of minutes, I said I would cover evolution. I don't have much time left, so I just want to say that doppelganger peptides are really neat systems or model systems to study gene innovation, particularly those expressed in snail hunting cone snails, because they're only very recently evolved. So their toxins are also very recent. And so we can trace the origin of these toxins because of their recent origin. So there are three general mechanisms by which these peptides typically evolve. So the first one is recruitment. And that means that there is already an endogenous neuropeptide. Let's say the cone snail itself makes an endogenous insulin, and that then gives rise to the venom counterpart through a process called recruitment. And Thomas Koch, together with Eivind Undheim at the University of Oslo, we recently submitted a paper on this very diverse molecular mechanisms of recruitments that happen in all these cone snail peptides. And then you could see some of these peptides, like shown here. You find the doppelganger peptide, but the organism itself doesn't have the endogenous neuropeptide. So it cannot have been recruited from an endogenous source. And those peptides evolved de novo or through horizontal gene transfer. And there are examples within this space where people have shown this. But just by looking at the figure, you could imagine that even though people have not specifically studied the origin of this peptide, it must have been by de novo or horizontal gene transfer. And then there's one notable exception, which is short peptides, such as RFA-mites, for example, in cone snails that just have an arginine phenylalanine motif, and opioid-like peptides can evolve de novo, so they can be in this space here, although there is an endogenous neuropeptide that could have given rise to them. And this is because it doesn't take much evolution to come up with 2 or 3 amino acids. And so you don't rely on your endogenous neuropeptide to give rise to these peptides. So really interesting evolution going on with these doppelganger peptides. So I'll very quickly show you one other study in this slide that we have done. So in some cases, when you find the doppelganger peptide first, and then when you trace the evolutionary origin of the doppelganger peptides, you can find a new endogenous neuropeptide. And there had been anecdotal case studies like this, but we decided to do this more systematically. So we published a study kind of recently where we systematically applied this approach and discovered various new neuropeptides in cone snails and other protostome phyla. And nobody wanted to publish this study, which got us really upset. But this has led to an industry collaboration with Novo Nordisk, which has been really fantastic. Okay, so with that, I want to say that venoms and poisons are one interest area in our lab, but we've also started to work on certain parasites and also are looking at sexual interactions for the discovery of metabolic doppelganger peptides. These are the people that have done all the work. Bea Ramiro, who used to be a PhD student in my lab, she's now in Ivan's lab, and she discovered this somatostatin-like toxins. Walden Yoshimoto, who is pushing the development of the pain somatostatin-like toxin. Ho-Yan Jung, who's just a pharmacological powerhouse. She recently returned to the University of Copenhagen, and she discovered the somatostatin-like doppelganger in Conus geographus. And Thomas Koch, who is a rising star in the toxin field and neuropeptide field, and I'm sure you'll hear his name many more times. And he's also just joined Ivan's lab at the University of Oslo. Various collaborators, people that have done illustrations, funding agencies. And I can no longer, being in the US, say anything political, but I thought I'd show you my lab before January 25 and after January 25. So people are leaving the US. And so if any of you is brave enough to come to the US, we have an open postdoc position on doppelganger peptides from amphibians. So please let me know. And with that, I'd like to thank you for your attention.